First of all, I would like to thank the organizers of the conference for providing the opportunity to present a study that our research team has recently published. And during my presentation, I would like to cover 5 points. First, I will give you a short overview about what the microbiota research group at the University of Applied Science in Kalisz is doing. Then I will spend some minutes on discussing what are the functions of a healthy gut microbiota and what are the consequences when the gut microbiota will be disturbed. Thereafter, I will emphasize on one particular role of the gut microbiota, namely the colonization resistance, and how this colonization resistance can be supported by the administration of products containing probiotic microorganisms. Then I will present our study and the results which we have obtained. And finally, I would like to give you an overview on how this study is fitting into our broader infantile colic research program in Kalisz. There will be also an outlook into what we are planning to do in the future. So what are we doing in Kalisz? And I think our moderator was so nice to introduce already a little bit my history. As you can hear from my strange accent, I'm German by origin, and I'm now working at a Polish university, but that is a complex story. So what our research team is interested in are probiotics and symbiotics. So, and most of these products are not pharmaceutical products, but they are food supplements according to the regulations in Europe. Our interest is especially in what role these products can play in our healthcare environment. And as you can see on the slide, our work is currently focusing on 4 areas. All of these areas are acute problems in our healthcare environment. So we have a program focusing on persistent Salmonella typhimurium, a program on infections caused by Klebsiella pneumonia or Clostridium difficile. All these pathogenic bacteria are causing significant problems for physicians around us. In addition, we are cooperating with pediatricians in the area of infantile colic. And for those of you interested in details of our work, a couple of our studies are available as open access publications. So just Google our research in the internet. Everything is available free of charge, and you can easily download our publications. So what is a gut microbiota, and why is the gut microbiota so important? The number of bacteria living in our gut is about 10 times larger than the number of cells of the human body. And a healthy gut microbiota is characterized by a high variety of different bacteria species. How many different bacteria species are living in our gut differs from study to study. However, I think it's fair to assume that there are between 500 to 1,000 different bacteria species colonizing our gut. These bacteria are accounting for about 1.5 kilograms of our body weight. So looking at these facts results in the question, why have every one of us invited 1.5 kilograms of potentially dangerous bacteria to live in our body? And I think the answer is given on this slide. We have invited these bacteria because they are doing a lot of very important things for us. They help us to digest what we eat. They are producing certain vitamins, and they are inhibiting the growth of pathogenic bacteria that can be found in our gut. And they are extremely important for the maturation and the regulation of the human immune system. However, the gut microbiota can become disturbed in a number of different situations. And there is a growing number of diseases for which it has been shown that patients have an abnormal gut microbiota composition. The gut microbiota can also be disturbed by stress and when we are becoming older. In addition, the gut microbiota is disturbed by healthcare interventions. Whenever an antibiotic is taken, this antibiotic will not only fight an infection somewhere in the body, but it will also affect the population of bacteria living in the gut. In some patients, this will cause symptoms, in some not. However, the disturbed microbiota will have consequences in all patients. And this we should never forget when we are treating patients with antibiotics. Other pharmaceutical interventions, for example, the therapy with proton pump inhibitors or certain cancer therapies, are also known to change the bacterial composition in the gut. As we see, the disturbance of the gut microbiota is quite often caused by healthcare interventions. And therefore, one of our teaching objectives at the university is to make our medical students aware of the fact that some of their future actions can have some undesired consequences on the gut microbiota. And hopefully this awareness will provide them with an opportunity to avoid or limit negative consequences related to this change in the microbiota for the health of their patients. So in patients with a disturbed microbiota, we might observe digestion problems, the lack of certain vitamins, the loss of colonization resistance, and a reduced immunity. Our team is very interested in investigating the possibility of supporting the colonization resistance of patients with a disturbed gut microbiota by the administration of products containing probiotic microorganisms. So, what is colonization resistance? You see a schematic cross-section through the gut. And under normal circumstances, the gut is populated by a variety of beneficial bacteria. However, in most of us, we would also find some pathogenic bacteria. For example, some Clostridium difficile bacteria. Luckily, the proliferation of these pathogenic bacteria is normally inhibited by the beneficial bacteria of the gut. And this is what is called a colonization resistance. How are the beneficial bacteria inhibiting pathogenic bacteria? I think this is a topic for another long lecture, but it's important to realize two facts. First of all, that different beneficial bacteria are using different mechanisms for inhibiting the growth of pathogenic bacteria. And secondly, that sometimes several beneficial bacteria join forces and act synergistically. So let us assume that a patient is taking an antibiotic. And for the purpose of this discussion, I have selected the antibiotic clindamycin. This antibiotic is quite commonly used, and especially dentists are liking this antibiotic because it is treating infections in the bone. And when the dentist is doing an implantation, then he is using clindamycin either for treating an infection or as a prophylactic measure. This antibiotic has a nice broad spectrum of antibacterial activity, however, with some minor but important gaps. And when the patient is taking clindamycin, a lot of the beneficial bacteria in the gut will be destroyed. While clindamycin is killing a lot of the beneficial bacteria in the gut, it is not killing Clostridium difficile, as this is a small but important gap in its spectrum of antibacterial activities. So, as Clostridium difficile will not be killed by clindamycin, and on the other hand, it will not any longer be inhibited by the beneficial bacteria because they are all dead. As a result, Clostridium difficile will start to proliferate. And this proliferation of a pathogenic bacterium can result in an overgrowth of the gut. And this, in some patients, can result in the development of a quite serious disease called pseudomembranous colitis. And this can have a fatal consequence for some of the patients. So the purpose of our study was to investigate if certain products containing probiotic microorganisms might have the potential to support the colonization resistance of the gut microbiota. And as we all know, there is a huge number of different products containing probiotic microorganisms available in the market. These products belong to different product categories. So we can differentiate between products containing probiotic yeast and those containing probiotic bacteria. Then there are monostrain probiotics containing only one strain of microorganism, and the multi-strain products containing several probiotics. Finally, there are symbiotic products which contain, in addition to the probiotics, a prebiotic component. A prebiotic is a substance that can serve as a source of energy for the probiotic microorganisms. A commonly used prebiotic is, for example, fructooligosaccharides. For our study, we have selected those products which are used in our healthcare environment. And in other healthcare environments, these products might look a little bit differently. So the products that we have investigated belong to 4 product categories. One of the products is a yeast probiotic containing Saccharomyces boulardii. 2 of the other products used by our physicians are bacterial monostrain products. One contains Lactobacillus rhamnosus GG, the other Lactobacillus reuteri DSM 17938. Then there is a multistrain probiotic containing 3 different strains of Lactobacillus rhamnosus. And finally, a multistrain symbiotic is popular with our doctors, which contains 6 lactobacilli, 3 bifidobacteria, and fructooligosaccharides. So in our study, we have addressed 2 questions. First, do these products inhibit the in vitro growth of certain pathogens? And secondly, are there significant differences in their inhibitory actions? So what pathogens have we investigated? For our study, we have selected bacterial pathogens, which are, unfortunately, I have to say, problems in our healthcare environment. Tested pathogens were Escherichia coli EPEC, which is an enteropathogenic Escherichia coli strain causing diarrhea in children, Shigella sonii, Salmonella typhimurium, Klebsiella pneumonia, and Clostridium difficile. We investigated the inhibition of these pathogens by using in vitro growth inhibition experiments. These are quite classical, old kind of experiments. But for those of you who are not familiar with these kind of experiments, let me briefly describe how these experiments were performed. In the first step, a petri dish was filled with a Columbia agar. This agar contains all what bacteria need to grow. Then a solution containing the test pathogen, for example, Escherichia coli EPEC, was put on top of this agar. Thereafter, the petri dish was incubated for 24 hours at 37 degrees. And during this incubation period, the pathogenic bacteria formed a dense floor on the top of the agar. In the next step, filter papers were soaked with the solution of our test products, and each filter paper was soaked with a solution containing 100 million of colony-forming units of the respective test product. The filter papers were then put on top of the pathogen culture in the petri dish, and the dish was incubated again for 24 hours at 37 degrees. During this time, the microorganisms in the test products formed inhibition zones around the filter papers. And the larger the inhibition zone, the stronger was the inhibitory effect of the tested product. The results of our experiments are summarized in this graph. As can be seen, the pathogens were all inhibited by the tested products. However, the extent of the inhibition by the individual products varied quite significantly. The weakest inhibition that we have found in our experiments were caused by the yeast probiotic and the bacterial probiotic containing Lactobacillus rhamnosus GG. Medium-sized inhibitions were observed for the Lactobacillus reuteri product and the multi-strain probiotic containing 3 different Lactobacilli rhamnosus strains. The strongest inhibition was found for the multi-strain Synbiotic. From the results of our study, we have drawn the following conclusions. Firstly, products containing probiotic microorganisms differ significantly in their in vitro pathogen growth inhibitory properties. Secondly, the tested multi-strain Synbiotic product caused the strongest inhibitions of all 5 pathogens investigated. And thirdly, in case the strong pathogen inhibition is a therapeutic objective, the selection of the right product might be of importance. For those of you who want to learn more about the details of our study, the publication is available as an open access under the download link given on this slide. So who is interested, please drop me an email. I can provide the link to you by email. That makes it easier to download. So in the final part of my presentation, I would like to put the presented study into the context of our infantile colic research program in Kalisz. The in vitro pathogen inhibition study was actually triggered by requests from pediatricians of our healthcare environment who are facing the problem of infantile colic in their daily routine. And these pediatricians are using different products. And anecdotally, they had told us that this multi-strain Synbiotic was in their hand the best product, but this was only anecdotal, and there was no really supporting evidence for this observation by the practicing doctors. As not all of you might be familiar with infantile colic, let me provide you with some basic information about this problem. Infantile colic is diagnosed based on the rule of 3, which has been first described by Wessel in 1954. By this rule, infantile colic is diagnosed in infants who are crying for more than 3 hours per day, for more than 3 days per week, for more than 3 weeks. Infantile colic occurs in about 20% of all infants during the first months of the life. In the literature, occurrence rates up to 40% can be found, and infantile colic is a major burden for infants, their parents, and healthcare providers. It is actually the leading cause why parents consult healthcare professionals during early infancy. Despite many years of research, the etiology of infantile colic remains unclear. There are a number of causes discussed in the scientific literature, among them hormonal, neurodevelopmental, psychosocial, and gastrointestinal causes. As you can imagine, our interest in infantile colic is related mainly to its potential gastrointestinal causes. In the recent years, there is growing evidence that infantile colic is related to a disturbed gut microbiota. In a number of publications, it has been described that the gut microbiota of Kolitsky babies differs significantly from that of non-Kolitsky babies. In addition, it has been found that symptomatic improvements can be achieved by the administration of certain products containing probiotic bacteria. So to better understand how pediatricians are currently addressing the problem of infantile colic in their daily practice, we have performed surveys among German and Polish pediatricians. At the moment, we have finished the data collection in Germany and are in the final phase of data collection in Poland. And on this slide and the next slide, I show you some yet unpublished results from our surveys among German pediatricians. And as you can see, more than 70% of the German pediatricians perceive infantile colic as high or very high burden for the parents. Not being able to calm down a crying baby is extremely frustrating for the parents. Therefore, it is not a surprise that infantile colic is related to maternal depression and premature termination of breastfeeding. The maternal depression is a big problem, and we don't like to see that breastfeeding is terminated too early. As we know, it is very important for the development of the babies. Infantile colic is also the major risk factor for the shaken baby syndrome. And in cases of the shaken baby syndrome, the baby is actually killed by a parent by extensive shaking. Shaken baby syndrome is not quite common, but every case is a tragedy of its own. So how are German pediatricians addressing infantile colic? As can be seen by a variety of measures, and often even with a combination of different approaches. Pharmacological interventions are frequently using probiotic or symbiotic preparations. Pediatricians are also using simeticone, a surface active compound, which is quite frequently used. This is interesting, as according to the literature, the efficacy of simeticone in infantile colic is more than questionable. Nevertheless, it is quite frequently used for the treatment of Kolitsky babies. And we interpret this as an indicator that there is a huge pressure by the parents on the pediatricians to help them and prescribe something. So what is probably also interesting on this slide is that you see a very small number of doctors who are not doing anything with these problems. So it is a major burden, not only for the parents, not only for the infants, but also for the pediatricians who have to do something. Based on this information from the in vitro pathogen inhibition study and the data that we have collected from our survey program among pediatricians, we decided to perform a small clinical trial. And in this trial, we have investigated the effects of the multi-strain symbiotic compared to simeticone in Kolitsky babies. We just recently have finished this trial, and at present the manuscript is in the review process for publication. So in the future, we would like to do one other clinical trial, and in this trial we plan to investigate the effect of the multi-strain symbiotic on calprotectin level in infantile colic patients. Calprotectin is a gut inflammation marker and has been shown to be elevated in Kolitsky babies. This project is still in the planning phase, and currently our main focus with this part of our research program is to find the funding which we will need to do the calprotectin measurements. So in case someone in the audience would like to support us, please feel free to contact me. With this, I'm coming to the end of this session. I hope I have addressed all points of my agenda and that there was some interesting information for you in the audience. The gut microbiota is and will remain a very interesting subject for research. And I'm very confident that research in this area is important and will contribute to better treatment of patients now and in the future. So I think we can now address some questions, but also feel free to drop me just questions by email. I'm also happy to receive comments and suggestions. Everything will help us to improve our research efforts. Thanks a lot. It was a pleasure to present. I'm looking forward to your questions.